Our next speaker is Dr. Lauren Aleksunes, who is the lead PI for the Transporter Elucidation Network and a professor of pharmacology and toxicology at the Ernest School of Pharmacy at Rutgers.
Thank you so much, Sarah. It's still good morning for a couple more minutes, but we're going to go through this quickly and the next couple speakers, and we'll get everybody to lunch, but it's been a fantastic morning. So I'm really excited to share a little bit about our work here today. We're championing studying placental transporters using an array of approaches, but one of them being through birth cohorts. So we have our topic today that is going to be placenta prostaglandin transporter, and what we are looking at this particular transporter is a possible early warning signal for exposures to environmental chemicals. And thankfully, our speakers earlier already kind of gave a preview of a couple of the chemicals I'll be talking about. Okay. So the transporter of interest today is the prostaglandin transporter. This is part of the solute carrier transporter family, or SLCs. They're involved in the uptake or influx of a lot of chemicals, both endogenous as well as exogenous chemicals. Specifically within this superfamily, we're talking about the organic anion transporting polypeptide OATP2A1. Now this one is a little bit different than other OATPs that are usually a little bit more promiscuous in their substrate profiles. This one is quite narrow and really focusing on inflammatory mediators, PGE2, PGF2, as well as some of the drugs that also resemble these inflammatory mediators. As you can see here, it is expressed very prominently on the surface of syncytiotrophoblasts throughout gestation. And so just let's take a step back and kind of talk about the importance of prostaglandins during pregnancy, and then kind of thinking about how changes in this transporter might be important. So on the left-hand side, I've kind of simulated a cartoon of the uterus and amnion. On the right-hand side, very simplified placenta in purple, and then in between, maternal lacuna, blood, extracellular space. So thinking about the fact that we're going to have, where's my animations? There we go. Prostaglandin synthesis is going to be occurring in a number of these different cells, including the syncytiotrophoblasts, as well as in the uterus and fetal membranes. Using cyclooxygenase 2, we'll have generation of those prostaglandins inside the cells. They're going to typically have to leave the cells in order to be able to act upon cell surface receptors in order to mediate a lot of their responses, so we can kind of see that disposition into these extracellular spaces. We then have the prostaglandins accumulating, and this is going to be important then in regulating the rupture of the fetal membranes as well as the contraction of the uterus. And so these are going to be two important steps in parturition. And so this is highly regulated by a lot of factors, but prostaglandin availability is going to be one of the key ones. So in order to have this fine-tuned homeostasis, we need to have other mechanisms beyond synthesis. And this is where some of these transporters are going to come in. Specifically, this prostaglandin transporter OATP2A1 is found on that syncytiotrophoblast layer, and it's going to be important in being able to regulate how much is available to this extracellular space, and then lead to the breakdown of these prostaglandins. And that's pretty much uniquely occurring in the placenta. And so we're very interested in kind of understanding this prostaglandin clearance and then associations with disease, but even maybe just with toxicities. So as we started looking at this particular transport in the placenta, we wanted to ask the question, what is known about its regulation in human placentas? And very quickly, going through the literature, and I'm not going to go through each of these papers, but just a few that I selected, there was a clear association that was being seen, is that in pregnancies that have preeclampsia, that their placentas have higher levels of this particular transporters. Now, actually, my hypothesis was that it was actually going to be opposite, that having lower levels would lead to less clearance from the extracellular space and maybe increasing risk of preterm birth, which has been associated in subsets of patients with hypertensive disorders of pregnancy. But the data are the data, and they're pretty clearly positively associated. So we wanted to also take a step back. As I mentioned, I'm a toxicologist, and wanted to understand, well, if we remove the disease component here, is it potential for environmental exposures to be regulating this transporter in a way that might serve as an early warning system before the onset of disease? So we wanted to do this in the context of a birth cohort, which is the UPSIDE cohort run out of the University of Rochester by Tom O'Connor, my colleague Emily Barrett, shown here, as well as Richard Miller. And UPSIDE is a low-risk birth cohort where we can see participants that are going to be monitored longitudinally, prenatally, and postnatally. Relatively healthy group. We have predominantly non-Hispanic white and Black participants, an array of educational status, maternal age, and early BMI, both around 28. About a third of the group that is nulliparous. And again, very low smoking rate. This is self-reported under 8%, and also very low complication rate. And so again, these were all low-risk pregnancies. And so this has been a really great cohort to understand relationships in the context of normal healthy pregnancy, the importance of environmental exposures. So student Chengwei Jiang, she's a PharmD PhD student at Rutgers. She's been very interested in understanding two of the classes of chemicals we've heard about earlier today, toxic metals like cadmium, which can act to accumulate in the placenta almost like a sponge and reduce placental functions, leading to reduced growth of babies, as well as those forever chemicals, the per and polyalkyl fluoral substances, or PFAS, which are more long-lasting, persistent in the body. And what's been seen in the literature in a lot of epidemiological studies is a positive association between these chemicals and diseases like preeclampsia. But again, we wanted to kind of take out that disease part and just say, well, what about just these exposures and this particular transporter? And so our colleagues at the University of North Carolina have performed mass spec-based quantitation of this transporter for us within the UPSIDE cohort. And we can see a large variation in range of OATP2A1 levels across about 250 placentas in UPSIDE. So Chengwei has then gone and modeled a number of different associations, both looking at factors related to baby, mom, as well as these chemical exposures, in order for us to ultimately develop a predictive model that interrogates potential regulators of the prostaglandin transporter. What Chengwei saw is that in bivariate analyses, she was able to identify a number of factors that had associations with OATP2A1 levels, namely birth weight, gestational weight gain in mom, pregnancy complications in that handful that we had observed, as well as two of these chemicals, particularly PFHXS, one of those PFAS we heard about earlier, as well as cadmium levels. So Chengwei then worked to build a multivariate model to be able to look at associations or predictors of OATP2A1 levels in the placenta. On the left-hand side, we can see anything that was negatively associated with OATP2A1 levels, and on the right-hand side, positive associations after adjusting for all of these covariates. And so even though we have a very small number of individuals that went on to have pregnancy complications, this was interesting to see at least a positive association, if not statistically significant. But when we looked at these two environmental chemicals, even after adjusting for all of these other covariates, we could see positive associations between chemical exposure and levels of the prostaglandin transporter. And even when we remove that subset of cases that did have pregnancy complications, these associations still continue to be statistically significant. So we would like to posit that there's a positive association potentially between 2A1 and these environmental exposures, and this might reflect an early warning system of placental stress during pregnancy. With that, I'd like to thank our groups at Rutgers, Rochester, and UNC, as well as our NIH funding and the invitation to speak at MPRINT. Thank you, guys.